Hey everybody, welcome back for I guess a double episode of the Journal Club this month. We're having our Halloween special. This is my buddy back here, overworked grad student, and I'm Dr. Oliver Medvedik, along with everybody here with Leaf and Lifespan.io, presenting to you from, well, not so sunny New York City. It's kind of rainy out there. It's going to put a little damper on our Halloween parade that we have over in the village every year. Haven't seen that in a while, but if you are in New York City, you should definitely check out the Halloween parade. And we're going to be covering a paper that involves, it's a very short paper. It's a PNAS paper that is looking at Drosophila lifespan, but it ties into a number of things that we've been discussing kind of over the many months here and years. Which is, how certain therapeutics are going to start to enter the market, and what sort of therapeutics will they be when we start applying them to human lifespan. And this is a paper that looks at drug combinations, and what they refer to in the paper. Let's see if I boxed it in here, but basically combination drug treatments where you can basically have a polypharmacological intervention. So basically using several different drugs that target several pathways all at once, and seeing if this effect could be better than having just one drug. And as you'll see in the paper, the answer is yes. It's an additive effect and perhaps slightly synergistic effect. And we saw some of this promising data in a C. elegans paper that we covered a while back. So this is a Drosophila paper that looks into this. So as time goes by, I think it'll be very promising to see these types of drug combinations employed in mammalian systems. And in fact, there was one such intervention. Those of you who were at our conference here in July, no doubt remember Greg Fahey's talk, where basically, is he at the Mayo Clinic? I can't recall if he was at the Mayo Clinic. But anyway, Dr. Greg Fahey did a basically a multi-pharmacological intervention looking at thymus regeneration in males, and they had some very promising clinical results. And I think they used a number of different compounds. I think they used it was growth hormone, and it was, I want to say DHEA, but there was a couple of other compounds that they used, and that showed very promising results. So having these types of polypharmacological interventions, it seems like that's a very promising avenue, and with all of this information that we have at hand right now, as far as having pathways that have been mapped, we can now use a rational approach to see which drug combinations might be the most promising. Yeah, so Keith says DHEA and 2 diabetes drugs. So it was probably, when you say a diabetes drug, probably it was probably metformin, I think. I think that's what it was. It was metformin. Okay. So without much further ado, let's share a paper here. So this is the paper. Triple drug combination targeting components of the nutrient sensing network maximizes longevity, by Castillo Kwan et al., involving universities over at the UK and Harvard Medical School here in Boston. So like I said, it's a pretty short paper. There's 2 figures. It's going to be a short one. So if you want to go trick-or-treating, we're not going to keep you here too long. But it's basically a Drosophila lifespan analysis. And here is figure 1. Is that coming in pretty clearly? Should I embiggen this figure? Shout out to Steve and Fatima behind the scenes.
Couldn't find my unmute button. No, it looks fine to me.
Okay.
Okay.
Right.
On this thing.
Okay, good. So basically, there is a picture here up in the upper left-hand corner where they basically show the pathways that they're going to be targeting. So they're essentially looking at the insulin like growth factor pathway. So so they're looking at basically 3 pathways. They're looking at the insulin-like signaling pathway. They're looking at, and the pathway itself bifurcates in two directions. So it essentially can influence FOXO and a number of other transcriptional regulators. And also in the other direction here, where we have another drug that's targeting a kinase, the mitogen activated protein kinase, MEK. Then there's also the mechanism of keep garbling the there's the mTOR pathway. Basically, they have mTORC1, but there's also mTORC2, and there's three drugs that target different components of these pathways. There's rapamycin, which is obviously targets TORC, which is target of rapamycin. There's a chemotherapeutic drug, trametinib, which targets MEK, which is mitogen activated protein kinase. And there's lithium, which you may or may not have heard of. Some of you have heard of lithium, obviously, as a therapy that's given to people who have severe depression. But there's a lot of data out there that's epidemiological that suggests low doses of lithium can actually extend healthy human lifespan. And I believe there's, they cite some of these in this paper here. There's a Japanese study that looks at lithium, and the dosages actually come from the environment. I believe it comes from, you know, what's naturally found in water. So this was an interesting study. And lithium is targeting a kinase here, SGG, which is the fly. I believe it's the fly equivalent of glycogen synthase kinase 3, GSK3. And interestingly here, downregulation of the insulin's sensing pathway or insulin-like signaling pathway basically can have a pleiotropic effect, meaning that by downregulating this pathway, you can release inhibition of FOXO, which is basically is a transcriptional factor that's involved in lifespan regulation and increases a lot of things, but basically it increases survivability. But it also, by downregulating this pathway, you upregulate some negative aspects, which can essentially downregulate things that are positive. So there's this pleiotropic effect that, interestingly, the authors want to suppress using lithium directly. So that's what they're assuming that lithium is doing here, which is a known suppressor of this kinase. Oops, I'm going to open up my chat here. Okay, sorry, that was from something else. Anyway, okay, so you have these three compounds here. These are all administered in the food of these flies. They also look at fly mutants, which there's peptides here that can regulate this signaling pathway. So these, so I'm just going to jump here and say these DILP, these peptides can attach to these insulin-like receptors, and by having these knockouts, you can extend fly lifespan. So this is a background. So this is a genetic background where you can essentially suppress the insulin sensing and signaling pathway here in fruit flies. By the way, one caveat to mention about when you study these insulin sensing and signaling pathways in organisms that are like C. elegans, fruit flies versus mammals, we tend to have, over evolutionary periods of time, pathways that basically regulate growth via the growth hormone, and pathways that regulate insulin sensing and signaling have sort of bifurcated into distinct pathways. And growth signaling and insulin signaling is sort of still through, the pathways have not diverged completely in these lower organisms. So that's something to keep in mind. Okay, so what do they do here? So one thing before they go into the lifespan, they note that taking rapamycin can have some deleterious effects, over chronically over a period of time. One thing is that it can induce excess levels of triglycerides in the blood, derived from the liver. So what they want to do here, so one thing that they want, and they attribute this through the activation of this pathway here that's in yellow, which they assume that lithium is going to suppress. And one way that they want to, show that lithium is perhaps, inhibiting this pathway. And they don't do this directly, so they're not showing lithium directly inhibiting the kinase here, right? So, obviously if you want to go down that rabbit hole a little bit with experimentation to determine that lithium is actually doing this, clearly we have to do some more molecular biology here. But they show that if they look at triglyceride levels in these flies, the control is just your normal flies. Lithium triglyceride levels are at 150 milligrams per, I guess that's per fly. I guess they have to control that for fly weight, right? I don't know how much variability there is between flies. But the levels go up in rapamycin, but then they come back down again if you have rapamycin and lithium, right? So one of their arguments here in this paper where they, the justifications for using these combination, approaches is that not only that you might get a synergistic or additive effect of lifespan extension by targeting multiple pathways, but they also say that they have the potential to reduce or the crosstalk might actually reduce side effects from one of the other drugs. So I don't know if there's any other evidence out there in the literature that suggests this, but this is something that the authors are purporting to show here, which I think is very very interesting. I don't recall any other paper that talks about kind of the crossover effect of reducing a side effect of another drug, right? So basically, what they're suggesting here is that the lithium is enabling, by having the suppression of this pathway here by your insulin sensing and signaling or insulin-like growth factor pathway, you get the positive effects of FOXO upregulation, but you suppress the negative effects of getting essentially the other pathway not activated. So anyway, on to the survival. Now, C is basically just an extension of B. So this is actually a survival assay, and fruit flies that have, it seems a little bit contradictory, right? Because, here something better is the flies are surviving less. And the reason is because in this survival assay, if they have more triglycerides released into their blood levels, obviously the flies are going to survive better because they're, the rapamycin is causing more triglycerides to be released. So you're starving the flies. They have more food to kind of, power their cells. And that's suppressed if you basically have rapamycin and lithium, right? So that correlates with figure B and C. Okay, so now they come to now we start getting into the interesting data here, which is looking at fly survival and in a number of different contexts. And here they're looking at the individual drugs themselves. So D is lithium, E is rapamycin, and F is trametinib, right? So they're each hitting these particular pathways, and they're looking at these drugs in the context of two fly backgrounds. The solid line, the dashed lines, are wild type flies, and the solid lines are these DILP235 essentially mutants, which are mutants for these peptides, which can activate the insulin and insulin-like growth factor receptor pathway. So essentially, you have a repression of this pathway. So that in of itself should lead to lifespan extension, right? So they're showing here, obviously, that if you have your control and you add your lithium, you have a slight extension. I believe in the paper, they attribute about an 11% extension or something around, a little bit more than 10% extension of lifespan average. I don't know if that's just average or maximal as well, but that's the figure given. And then if you have your, if you add your, in the context of these genetic mutants in some of these pathways, I think you get a slight extension. Lithium, you get a slight extension, which they attribute to basically the suppression of, or the activation of CNCC, another growth factor here, which is basically through the repression of a repressor. And that effect is not seen for rapamycin, although you do get an extension for your normal lifespan. in your controls. And here they say it's only evident for lithium, but not for the other two. But it looks like maybe in trametinib you get a slight average lifespan extension in these mutant backgrounds. But anyway, they attribute this modest but I believe reproducible extension because the lithium is suppressing this pathway here, which actually gets activated when you suppress everything downstream. So it'd be interesting to look a little bit deeper here to see if that is indeed what's taking place, because that would be very intriguing that you have lithium doing this and basically essentially blocking a side effect of this pathway that's being activated. Okay, I'm going to stop sharing here for a moment. Take a little breather. I don't notice any more questions here, but we're going to go on to the next figure 2, where they actually do triple drug combinations. And there they actually see a pretty robust response where the effects are at least additive for all 3 of these compounds. So I'm going to go back to the figure. Share this. Okay. There we go. I'm going to scroll down here. And so this is essentially the kind of the most important figure of the paper, figure 2A, which is they look at triple drug combinations which maximize longevity. It's a bit busy, but this is essentially, you can see that of all these colored lines here, right? The red line is way to the left here, and that is your triple drug combination. And the stars basically indicate that the statistical analyses that they did suggest that it's, that it's good. So rapamycin, trametinib, and lithium. So you notice that, control is down here. It's kind of hard to tease apart here, but basically, if you add double drug combinations, you get some additive effects. Rapamycin and trametinib is the next best, but you get an even better response if you combine all 3, rapamycin, trametinib, and lithium, which the authors say double combinations give you, if we dig through here, about a 30% increase compared to each compound alone, which averaged 11%, and they say that the median lifespan is extended by 48%. So at least an additive, perhaps a synergistic effect. Interestingly here, so this is basically the numbers of flies that they actually assayed, and I was just kind of just as an aside, I was trying to figure out how they're doing these experiments, and they say numbers in parentheses show the total number of flies over the number of sensors. So I was looking at these numbers and I'm like, what is a sensor? Like, I know what censorship is, and Steve suggested it was the thing that a priest waves around to, emit fumes of frankincense. And I was like, yeah, that's that too, but I don't think that's what they're doing here. So you have 274 flies that are alive, and then you got 7 sensors, and then 10 sensors. And then I started digging through papers, and there was an article on how to actually count flies when you're doing lifespan assays, and they said, yeah, but don't count the sensors. And that was it. And I'm like, well, that doesn't explain anything. They said count the dead flies, but don't count the sensors. And I'm like, still don't know what that is. Until I finally found a paper which basically said that what a sensor is either an escaped or accidentally dead fly. So basically, those are the flies that flew out of the experiment, or basically got accidentally smushed, maybe in the screw cap, and that is the technical term for a sensor. So I guess that sounds nicer than, it's like going to the hospital and saying, I'm sorry, but you're somebody you loved, he unfortunately died of an iatrogenic illness. And you're like, wow, that's pretty rough. And then you realize that iatrogenic illness just means that the hospital screwed up and they accidentally killed your loved one, right? That's basically what that means. So a sensor is basically a fly that got away. So they didn't count that in. So obviously you want to be careful with these numbers. You don't want to introduce statistical bias, but that is what a sensor is. So there you have it. Something I learned today. So those are the, so looks like whoever was doing the counting was doing a pretty good job of keeping track of flies. So, only 10 got away in this batch of 482, and only 3 got away or accidentally smushed in this batch of 444. So good job. All right. That is a bit of a technical aside. And then figure C, D, and E basically look at, if you've seen these in mouse studies, right? If you give a drug to a mouse, is the mouse actually eating the stuff, right? Or is it not effective because it's just spitting the food out, right? So I guess it would be a little harder to do that with flies. So they look at feeding behavior to show that the feeding behavior is about the same for every one of these drugs and these flies. And they do this test called a proboscis extension feeding behavior assay, which I'm assuming is measuring how many times the proboscis comes out and touches the food. I don't know how you would measure that, actually. So I'm sure there's a materials and methods paper out there that details exquisitely how you would measure the proboscis extension during this feeding behavior assay. I'm gonna open up the chat here. Oh, the gavage method on flies. Yeah, that would be, yeah, that could be maybe that's a practical joke in fruit fly labs, right? So the first grad student that comes in here, you give them a stack of fruit flies and say, okay, you're going to have to do oral gavage on these flies, and so go figure it out.
Could be a good Halloween prank, right?
And all the other grad students are like, he's doing oral gavage on the flies.
I'd be very impressed. Give that man a PhD if they manage to pull it off, though.
Yeah. But anyway, so the food intake, maybe, I don't know, I don't know if that's significant for trametinib, but the food intake and the systemic levels of the drug. So I guess they had to smush the flies later and measure the drug concentrations in the flies are about what they figured. So this is just ascertaining that whether or not the flies actually ingested the stuff, because if you have fruit flies, you basically mix it up with food and some kind of mush that's in the bottom of a tube, and you throw your flies in, so you're assuming that they're eating the stuff, right? So it's kind of hard to control that, so you have to make sure that they're ingesting the right amount of food. They also looked at fecundity, so a lot of lifespan assays. If you extend lifespan, it's known to basically reduce the fecundity of so there's a trade-off, and one of the trade-offs is less reproduction taking place, like less egg laying. Interestingly, you get this decrease with trametinib, and let's see, what's the colors here? Purple. God, I don't know what the colors mean here anymore. I think rapamycin is purple, green. Oh, no, that's not right. Well, anyway, I can't find what the colors mean, but they're representing the different drugs that the flies are taking. Interestingly here, the triple drug combination, so the trametinib, even though it's reducing the fecundity, these triple drug combinations are not reducing it any further. So there seems to be a kind of a plateau at the end. So it's not, basically the bottom line for the authors here is that any extension of lifespan that they're getting is not correlated with a further decrease in fecundity than an individual treatment, at least not with the trametinib. So when they give lithium and rapamycin, it's not getting any more severe than what is already kind of plateaued there. Okay, so I'm going to stop sharing there. So that is the paper. Very, very, I think a very nice, clear, direct paper. I think we're going to be seeing a lot more of these polypharmacological intervention studies taking place. Certainly we're already seeing that in clinical trials, like this trial with Greg Fahey, and also the trial with that we covered with senolytics, which I believe used, was it dasatinib and fisetin, or was it quercetin and fisetin, or maybe a combination of one of the two in two different papers? I can't remember.
It was D and Q.
D&Q, right? So I think we're going to start seeing more of these, especially now that we have a theoretical basis for doing so, right? So we now have compounds where it's known which, or strongly suggested which kinases are being targeted in the individual pathways. So we have now a theoretical basis for using these types of polypharmacological interventions, and it's promising to see that we're getting results that suggest that that's the way to go, both in early clinical trials and also in animal studies, such as in Drosophila. And I don't know if they've done that. Have they done these types of poly trials with mice? Many, maybe not.
Not really. We're planning to at MMTP.
Yeah. So anyway, so the bottom line is that this appears to be a very promising approach, right? So I don't know what else to say. Any comments out there? No, it's a little. Everybody's out trick or treating.
I think everybody's. Yeah, they are quiet. I think they're all blown away. But so great news if you're a fruit fly and you're not one of the sensors.
Yep. Don't be a sensor. Don't accidentally die or escape into the wilderness.
Yeah, that doesn't happen so much in the human studies. They don't tend to escape, or certainly don't fly away.
They tend to drop out for a variety of reasons. Well, I don't know. They do accidentally die, right? Yeah.
Oh, cheery thought.
Well, it is Halloween.
It is Halloween, and he is positively ghoulish this evening. So yeah, nice short to the point paper, interesting. Shows the potential of multiple drug combinations.
Mm-hmm.
But as with anything to do with combinations, the challenge is trying to discern whether it's an additive or a synergistic effect.
Yeah,
it's always difficult. But
so,
and I think it's going to be more common, as you say, because a lot of these interventions do require multiple targets and drugs to hit those targets. So they're going to have to come up with a good way of actually gauging, whether an effect, how and why an effect is occurring. So that's interesting.
Yeah. So, I mean, it's going to have to, people, I guess, people are going to have to accommodate this into their clinical trials, because I know it's hard to look at multifactorial approaches. But if the answer is multifactorial, then we best accommodate, those types of trials, because that's the whole point, isn't it? To have something that works. So I just want to reiterate from the paper that having this multifactorial or polypharmacological approach, basically the authors list 3 advantages. One being the obvious one, that you have an additive or synergistic effect. The other, as I mentioned, which I thought was intriguing, was reducing each other's side effects, which they cite a paper here. And I don't know how many papers there are for this is, I don't know how much evidence there is. There's this, I'm guessing there might not be a lot out there. I mean, besides this paper and some others that suggest that, like with the lithium, that you might counter a side effect from another, intervention that has a partially positive effect, but has some pleiotropic negative effect. And then the other thing that the authors mentioned, obviously, is the potential to counter resistance from feedback. Certainly you see that with, I don't know how much of a problem this would be with lifespan intervention drugs, but certainly with cancer chemotherapeutics, you take one type of small molecule and you can have a mutation. And certainly antivirals, that sort of thing, where things are dividing much quickly, you can get some sort of resistance. I don't know if there's actually any evidence of, that's another question I have, evidence of resistance from feedback. If you're taking a drug such as resveratrol, rapamycin. I don't know. You name some compound out there that you can take where basically the effect wears off. I don't know if there is such a potentiality for for at least the way these drugs are working for for lifespan enhancement, but that's something that the authors mention here. But anyway, a lot of very interesting points raised here, and good job.
Yeah, a fruit fly paper, which is unusual. Fly paper, as you called it.
I called it fly paper.
Fly paper. That's something you hang in the greenhouse to keep the flies off your tomatoes.
Yes.
For all you gardeners out there like me. So that gave me a chuckle when you said it was a fly paper.
It's fly paper. Yeah.
Big yellow sticky thing that hangs from the ceiling.
It means the same thing here as well.
See, some things translate, even if some of our language doesn't always.
Yeah.
But that's a topic for another time. We could have a few laughs at that. So anyway, you're off trick or treating, I believe.
Yeah, I'm still at work. This is Eastern time, so it's not 2:00 here quite yet. But it is kind of grim out there. It's overcast, drizzly, not so cold, but rainy out there.
I would say that was ideal for All Hallows Eve then.
Yeah, maybe. I mean, maybe it'll dry up a little bit. Maybe it'll be less rainy out there. But anyway, we haven't picked a paper yet for our next topic. There's a couple of papers that we've been bouncing around on between our emails. We'll look forward to that one.
There's quite a lot of stuff going on at the moment, really. I mean, we've got that mitochondrial unifying mechanism paper, and potentially the one of the researchers might join us. That could be interesting.
Right.
But there is a lot of stuff going on. I mean, you've got the senolytics, the trials that are coming on. I've just seen today that Unity Biotech is also now entering phase 2 for senolytics. That's, again, there's a lot going on. So
there's a paper paper came out with from Harvard and a bunch of other institutes showing correlation with hyperactive neuronal activity in certain regions and lifespan or inverse relationship, which there have been other papers which suggested overstimulation of certain areas that correlates with dementias or Alzheimer's. So that's an interesting paper as well.
Yeah, Lab's got another one out with telomeres. Mice with super long telomeres live longer. They've got a mouse strain now with even longer. I mean, mice have got very long telomeres anyway.
Yeah, but they degrade much quicker, right? So
they do, and it's the rate of the burn that dictates the lifespan, as they showed earlier this year. But they've made one now with super long telomeres that lives longer. Makes sense, really, because it's even though it's burning as fast as a normal mouse, it's burning down. There's more to burn down, so it's going to live longer.
Yeah. That papers studies like this are very interesting, because if you can essentially make I don't want to say an infinite telomere, but let's say you make a telomere where you don't have to worry about the telomere loss or attrition being the rate limiting step, and the mouse still ages and dies, then you could start to basically catalog what are the other what are the other age drivers, right? So if clearly if you have a mouse that still has pretty long telomeres when it kicks the bucket and is still suffering from aging, albeit at a later age, age-related disease manifestations, then telomeres are just one part of the things that are drivers of diseases of aging. So it'd be interesting to see that paper from that perspective.
Yeah, and then there's another one, David Sinclair and dozens of other important researchers as well. They've just published a paper about double-strand breaks and their contribution to aging. So they found the mechanism of DNA damage is basically causal. Double strand breaks are causal of epigenetic drift, and that's really significant because it ties genomic and epigenomic changes, i.e. those two primary hallmarks. It ties them together very strongly, and of course they're tied also with telomeres because that's part of the stability aspect, genomic via TPE. Wright and Shea talked about many years ago.
Right.
But the interesting thing about the paper was it kind of sort of backs up the hallmarks, and it builds on David's, I think he calls it the information loss theory of aging. I think it's the information loss. He talks about it in his new book, Lifespan, why we age and why we don't have to.
Yeah, I shipped those books out, but I helped ship them out, but I haven't gotten a copy myself. I talked to David recently on the phone. He said he was going to get me a copy. I'm gonna hold him to that.
Good book, apparently, folks. So if you're looking for a good read, quite accessible. Yeah. And if you visit our website, there's actually, if you look on the webinar section of our site, you can actually find the AMA with David recently, where he read some of the some passages from the book, and it sounded rather good. So it's not, you don't need to, it's not the kind of book where you need to have a PhD to understand it. It's very accessible. It's very readable. And I think that's super important, especially in disseminating information from the lab to everyday people on the street that who are not necessarily going to understand all the stuff, the jargon. But so I think David does a really good job of sort of cutting through that and translating it. So that paper was interesting because it kind of sort of says, hey, look, DNA, this is why DNA damage drives epigenetic changes, which of course then change what the cell thinks, does, and how old it thinks it is. So that's a very interesting paper for me personally. So I don't know. I think the writing's on the wall, it being a cause rather than a consequence. But some people would debate that. Perhaps we should have a debate about that sometime as a webinar. Epigenetics, cause or consequence. Probably turn into a fist fight at some point, right?
I don't know. Anyway, no comments from anybody, huh? Well, I guess it was to the point. We had everything on this paper.
Plenty of people watching, so they must have been entertained. So yeah,
good. Maybe we should do more of these shorter papers.
or do a couple of shorter ones per show.
Okay, so next month we will try to get one of the authors in the paper, right? The paper that I forgot the title of it.
That's all right. It's about 50 miles long, like most papers. I can't remember what it was. It was something to do with mitochondrial damage and the unifying mechanism for progerias.
So
something along those lines.
So so we'll aim to get one of the lead authors on as we did a few journal clubs ago, which I thought went really, really well, and we'll try to do that again. Try to reprise that for next month, which is tomorrow. But not journal club isn't tomorrow, but next month starts tomorrow.
Yeah, let's not do one too soon, Oliver. I mean, we're keen, but
okay.
So that's great, and obviously we will see you again probably towards the end of the month, same sort of time. We'll let you know what the topic will be, and also just a brief mention that the Mito Mouse campaign has done exceptionally well. The second stretch goal is shortly going to be completed thanks to longevity. So that means that the campaign has been, well, an overwhelming success. And thank you to everybody that donated to that project. I think we've got about a day and a half of it running still, and you can still donate if you're interested in getting a t-shirt or one of the various rewards. Check it out over at lifespan.io/MitoMouse. And as always, I'd like to thank the Lifespan Heroes, our monthly patrons, for making it possible to run this show and many, many other things that we do. And if you're interested in learning more about the heroes, you can learn more at lifespan.io/heroes. So that's great. We will see you next time, and enjoy your trick-or-treating, and don't eat too many toffee apples. They're not good for your teeth.
All right.
Like that anyway.
Happy Halloween, everybody.
Take care, everyone.